I'm Pauline Rudd. I'm the NIBRT Professor of Glycobiology at University College Dublin. And that means that I have two main sets of responsibilities. One is to maintain the high quality of the fundamental research that my glycobiology does. And the second is to work with the National Institute for Bioprocessing Research and Training, which is an initiative by the Irish government that supports the pharmaceutical industry in Ireland. So we have several facets to the research we do, but it all centres around the technology that we have. And over the years we've built up really world-class expertise in glycan analysis. Now, glycobiology is a field which stretches into every field of biology because more than 60% of proteins are glycosylated molecules. So that means that you can be interested in any field from development to immunology to neuronal diseases because they all involve sugars at some point. The most research we've done recently has been in the area of cancer because it's very well established that glycosylation of proteins changes during tumorigenesis. And so by monitoring those changes we can distinguish different stages of cancer, different responses to therapy. And so that's a very important part of the work that we do. When you see changes in cancer, one of the first questions you ask is, how did that change in the sugar pattern happen? And what's the effect in the disease? Does it contribute to disease pathogenesis? So that leads us into understanding glycosylation pathways, how the sugars are made, what happens in cancer cells that makes them change, and why a cancer cell that makes that change has an advantage in terms of metastasis. And all of that information, of course, then translates into the work we do with bioprocessing because we're trying to understand what changes glycosylation processing in making biotherapeutics. And I think everybody who works in the field realises it's very difficult to control glycosylation. So the more we understand about the factors that control the process, then the easier it is to get a stable situation in your bioprocessing plant. The pharmaceutical companies come to NIBRT because our analytical capability is such that we can analyse the glycosylation of their therapeutic. When the drug starts to be made in bioprocessing plant, you need to be able to track it so you can make sure that the glycosylation isn't going wrong during the process. And we also need analytical techniques to give the kind of information that the regulators need, which is what percentage of glycans do you have that are related to safety and efficacy. And then finally there's product storage. We have to have a method to check that the glycosylation hasn't changed during storage. So all in all, you need a lot of different techniques depending on where you are in this process. The vast majority of the new protein drugs are glycosylated molecules, and the most burgeoning section of those is the development of monoclonal antibodies. Once you understand the sugars are really important, then the next thing is to understand which sugars, because sugars are built of different building blocks, and you need to get the right building blocks for the function that you have. It was really only when erythropoietin began to be used as a drug that people understood the importance of the glycosylation for the PK, for the pharmacokinetics of the drug in the patient. So in that case, if you have the right sugars on, the drug is in the patient for 3 hours when it stimulates the production of red blood cells, which is part of the reason it's used for treating anemia, for example. But if the sugars are wrong, if they don't have sialic acid on, it leaves the patient in 3 minutes. So it has no time to perform any stimulation of the red blood cell development. A couple of years ago they brought out a much more serious guideline which says that antibody glycosylation in particular has to be well defined. You have to report what are known to be critical features in relation to the way that drug will act in a patient. So you have to report the levels of galactose that are there, the levels of core fucosylation, the levels of sialic acid, because we know all these features affect the way that this drug will behave in a patient. The requirement to do more detailed glycosylation is getting heavier, and particularly because if you're making a biosimilar, you're trying to make the same molecules. The requirements for a biosimilar are actually very stringent, actually. In some ways it's more easy to be an innovator than to try and make a product which is the same as one somebody else has made. We needed a very robust technology that didn't require huge numbers of years of training to be able to interpret the data. We also needed something that was very sensitive, that had a good range, so we could identify, say, 0.5% of the total glycans, and we still had good signal to noise. We also needed something that's quantitative, and that's really vital for the research work that we do. Quantitation is vital, and it's also really important for the pharmaceutical company as well, because you don't just want to know you've lost fucose, you want to know how much have you lost. We have two labs really. We have our research labs, where I think we have 16 alliances, UPLCs, HPLs, H-class. So it's a very impressive suite of Waters instruments. Then we also have a mass spectrometry suite, so I think we have 7 mass spectrometers, most of which are Waters instruments. You know, first and foremost, it's the absolute reliability of the instruments. And secondly, I think it's the ability to communicate with Waters not only at the technical level, so the service engineers who are obviously superb, but also with the people who are really understanding the science that underlies the separations that you're trying to do. If you think how much funding has gone into genomics and proteomics, glycomics by comparison has had far less funding, and I think now the attention needs to turn not only to glycomics but to other post-translational modifications, and see if we can just get information on a big scale that will allow people to look at the whole systems, because the future for me is in systems biology, to understand how all these different things that are going on in the cell are related to each other.